Thanks for invitation. Thank you for nice introduction. Really glad to be here, to be able to be here in person. That we are kind of not used to that anymore so much. So what I'm going to be talking about today is really kind of in between. Even though my general interests are both, or interests of my lab really, are somewhere between the structure of the genotype-phenotype maps and this a little bit more theoretical parts of how genotype-phenotype map, for example, pleiotropy, for example, epistasis, what consequences they have for variation and evolvability. But a lot of what we do has to do really with reproductive biology, and I'll show you one study today that not really very strongly, but already starts connecting these things. Yeah, this is my title. So, no preterm birth at the cost of endometriosis, antagonistic pleiotropy of a SNP in a human reproductive traits. So the way how this study was really started was with a quite simple question, and that was, what mechanisms determine human pregnancy length? You might think that we know that, and we actually don't. We don't know what determines when women go to labor. We can describe it very well, but we don't really know what starts it. And for many other mammals, we don't know. For some, we do. That's of course very relevant, because I was working in medical school for 6 years prior to coming here, as Christian already mentioned. And one of the big questions, and one of the reasons why I was hired there, was really to try to understand why humans have so much of the preterm birth. So preterm birth below 37 weeks of gestation is of course a very damaging factor. So trying to understand what actually determines the gestation length is a big question. And of course, just from the genetic perspective, we have two parts, right? We have the maternal side with maternal genome. We have a fetal side with both fetal and maternal and paternal parts in there. And what we do know a little bit about, just from the statistical analysis, is that a big part of heritability seems to be running on the maternal side. So there is an idea that probably the genetic factors that determine the preterm birth might be in the maternal biology. So let me just give you a very quick run through human reproductive biology, the parts that are necessary. One really important thing is that females cycle. That means that a lot of biology, actually, of female biology, has a very strong temporal component, so very strong temporal heterogeneity. Of course, the important part here, ovary, during the cycle, we have the follicle development and ovulation, of course, and then the production of progesterone in corpus luteum. This whole thing is controlled and regulated really as a dialogue between the hypothalamus and the ovary, by the endopituitary in between. Estrogen and progesterone as really important hormones, but not only, also the luteinizing hormone, and of course a suite of others. And also important is that this dynamics not only has effects in the ovary and hormonal effects. These have, of course, oops, here we go. Sorry, too much. These have, of course, also effects on the dynamics in other tissues. And in particular, what we will be talking about is of course, the uterine tissue. So what you see here in the correlated with the hormonal dynamics, you see the strong proliferation of uterine tissue in preparation for implantation after ovulation, and in humans that would mean, of course, at the end, the shedding of the tissue and the menstruation. The really important part within the uterus that happens is that the endometrial stromal fibroblasts, in particular, these are here shown in cell culture. They transform themselves into decidualized cells that look much more. So these are fibroblasts, and they look here transformed much more, almost like epithelial-like. They are the ones that form the interface between the uterus and the placenta during pregnancy in most mammals. In humans, they actually transform themselves even in non-pregnant cycle. So that will become important. All right. Okay. So what did we do? We ran a GWAS study. This is already published, so I'm not really gonna speak very much about this GWAS study, more about the functional genetic analysis following it. It was a really big one, about 43,000 individuals. So we used the data of 23andMe, and it was then checked still in another cohort of about 9,000. This is mostly the European ancestry, and really what is important for me here to say is that this study was run on continuous data, so really on gestational length, not asking just a question preterm or not. Okay, it's important because that's actually a trait. So we have, I'm focusing on one locus in this presentation, and that's the Wind4, the polymorphism in Wind4 that we detected, among others. All right. Okay. There are actually several variants, and of course, the first thing that one has to go about is try to figure out which of the variants in this general locus is actually probably responsible for the signal that we get. And we very soon started looking at a particular variant, and I will tell you what gave us the idea. But of course, these are the questions that come up once you have one association, right? You would like to know which locus is actually causally associated with the phenotype. You of course have the question which gene is affected. Of course, these variants are practically all in regulatory regions. Then you might want to know in which spatio-temporal context does it work. That's a hard one. What does it do? And of course, if you're looking into different model animals, then you want to know whether you can actually translate that into humans, right? So the arrows here just practically say that, of course, it's sometimes hard to answer some questions if you haven't before answered the others, but that's sometimes a little bit circular, right? So it's hard to figure out which context if you don't know which gene you should look at, right? So what comes in addition to this study is that the locus I'm looking at, the general locus, has been associated with multiple different diseases. It's been associated with susceptibility for invasive epithelial ovarian cancer, the endometriosis with uterine fibroids, and also with breast cancer. And so what I just wanted to just quickly explain what endometriosis is. because it will become important. We were talking about the shedding of the tissue, of the decidualized tissue at the end of the cycle, right, of the proliferated decidual cells. These cells, when they detach, they can flow both out of the vagina. They can also flow backwards through the fallopian tubes, and they always do. So that this itself is not disease. Um, but what happens with endometriosis is that they can attach again. And then so in the peritoneum, on the outer side of the fallopian tubes, around the ovaries, and they can actually react then to estrogen in cycles, grow, and that's very painful. So that's what endometriosis is. What's interesting about this looking at these additional associations with this locus is that all of these traits are really estrogen responsive tissues. So defects in estrogen responsive tissues. So that was one hint. But when we looked at how they affect, what direction the association goes, we noticed that the minor allele at this locus is actually deleterious for those associations, but is protective against preterm birth. So the effect goes in the different directions. That's why I call it the antagonistic pleiotropy. I'm sure that you probably have known about antagonistic pleiotropy before, so just a quick run through. We have a mutation with advantageous effects on some traits and disadvantageous on others. The original idea was in trying to explain the evolution of senescence, where we have positive effect on reproduction, but then aging in later stages. It's very often put into context with cancer-associated alleles. And of course, one of the consequences is that it, due to positive and negative effects, or due to positive effects, the alleles that cause negative effects are kept at relatively high frequencies in populations, right? There's always a question, of course, with pleiotropy in general, whether it's due to some direct combination of direct and completely indirect effects, or whether it's really the same mechanism that is in place in generating both traits, and we'll try to answer the question. One really interesting aspect of this particular locus is if we look at the distribution of the frequencies of these two alleles at the locus, we noticed that it actually varies quite a bit across the populations. And interestingly, not only these frequencies vary quite a bit, but also both endometriosis and the frequency of preterm birth vary accordingly. So I've just shown here two extreme populations in Africa, where it's practically the C allele here is practically only the major allele, so to 99%, and in East Asia, where we have half and half, and in East Asia it's really it's one of the highest frequencies of endometriosis, whereas in Africa we have one of the highest frequencies of incidences of preterm birth. All right. Of course, I don't think that it's one locus that sorts this out, but it's still an interesting pattern that we see. All right. Okay. So another reason why we are focusing on one particular locus, on one particular SNP in this region, is that we could associate it with a really improved binding of estrogen receptor. So estrogen signals through the estrogen receptor. Estrogen receptor is a nuclear receptor. What it means is it binds the estrogen, it goes to the nucleus, and in this case, it actually dimerizes, it goes to the nucleus and actually binds to the estrogen response element and works like a transcription factor to regulate very many genes. Matt Weihrauch has helped us a little bit with this prediction. So we could identify here the such binding site of the estrogen receptor, and this is our mutation. So the T actually generates a fantastic binding site of estrogen receptor just in the first intron of the Wnt4 gene. And here is just you can see that that in the particular decidual stromal fibroblast that is actually an open chromatin region, and this is really the Wnt4 gene. All right, we tested that experimentally whether it binds. So this is the electrophoretic mobility shift assay, where you have in different lanes the two different alleles, so the probes of the two different alleles, and then you add either the receptor or the lysate of the particular cells, and then at the end the antibodies. You can see the different different bands of the two alleles. The T is the mutant allele, and the C is the other one. And then if you add to that also the estrogen receptor 1 antibody, you see that there is a shift. And so we identified the right binding of the right thing, and it's differential between the alleles. All right. Okay. So of course the next question is, yeah, sure, it's in the first intron, but we know that this regulation can go a long way away from the actual actual gene. So we started, of course, looking for a model that we could use in order to figure out what this mutation might be doing actually really in vivo. And you see this part is actually highly conserved, and so we could very nicely take the mouse. Of course, also the location around it is quite conserved. Wnt genes, of course, have very many functions and are highly conserved across mammals. So what we did was we generated transgenic mice, and we generated 3 lines, but this one was actually a deletion, and these 2 had the right single nucleotide substitution. Most of the stuff that I will be showing you is comparison between the homozygotes, and most is done in Ki1, okay, in the first line. We confirmed at least the major effect also in Ki2 though. All right, so these are the 2. The mice that we took were C57 black. Oops, sorry. Okay, so the first thing that we looked, of course, at was the general phenotype, the overall phenotype, and there is actually no effect on the mouse gestation length and or on the litter size. So unfortunately, not here. But perhaps that's not such a surprise, given that of course this is just one of the loci, and on the other hand, pregnancy between mice and humans is quite different, and I will come to that later on. And of course, then we started to think where would we have to look at to see which gene that would be. Of course, the uterus is the first choice. but at what time, given that there is a cyclicity, that there is such a strong heterogeneity between the phases, and since this is the effect, downstream effect of the estrogen, the time of choice would be around the peak of the estrogen in the cycle, and that's at the late proestrus that it happens. This is the peak of estrogen in the females that actually triggers ovulation. Okay, so so we started looking at proestrus and estrus, and started looking at the flanking genes, of course. And what we see here is the actual effect that we find. It's at the time of estrogen, the Wind4 actually increases in expression. It might seem little here, but it's actually four times. And the other flanking gene, that's the CDC42, that has been previously thought to be the target of the mutation, doesn't seem to actually have an effect. All right. We also looked at the pregnancy at 7.5 days post-copulation, and looked at both of these genes, flanking genes Wind4 and CDC42, and you see that at that time Wind4 is quite expressed, but it's not differentially expressed between the two. So it doesn't seem to change the expression of the mutation doesn't seem to change the expression during the pregnancy. Yeah, that's a little bit harder to see here. We then of course started asking, okay, so this is gene expression in uterus, but where in uterus? What cells are expressing really this this Wind4? And this is RNA scope, so in situ. And we have on the right side here in red the uteri of the transgenic line, and on the left side of the wild type. And what you see, or maybe don't see so well, is so this would be epithelium, luminal epithelium of the uterus, and here the glands. In both, we have the expression in luminal epithelium, but in the transgenic lines, we also have strongest expression in stroma. So endometrial stromal cells are probably the ones producing the higher levels of Wind4. This is a little bit closer so that you can see the difference a little bit better. Okay. And we went a little bit further still and actually isolated the cells from the tissue, specifically the endometrial stromal fibroblasts. And now when our sample, of course, is enriched, the difference between the wild type and the transgenic mice is much higher. Okay. All right. There are other phenotypes that we find during estrus. So one phase later, just the next phase, these are the cross sections of the uteri. This is all non-pregnant, of course. The cross sections are much higher. This is the transgenic. This is the wild type. And even with relatively low number, we get quite some significance here. All right. Okay. So it seems we might have a spatio-temporal context in which it works, but of course one asks, so okay, it's Wind4. Does it matter? Does it do anything to the system? And what is it? And so one way to ask this question, of course, is to go for an RNA-seq and see what other genes might be regulated by that. What might that mutation actually really do? And you see, there's quite some dysregulation that we find in RNA-seq. this is proestrus uteri, RNA-seq, and always go to, just to take this apart a little bit, we have a down regulation of more than just these 4 genes, but these are the kind of the strongest here. That seems to imply the down regulation of the proliferation on the one hand, and then we have upregulation of quite some genes, and that seems to be genes associated with decidualization, the process I told you about already. Although you might think, but in mice they don't actually decidualize non-pregnant, so that's a little bit of an interesting conundrum. We have glucose utilization and invasibility, and that's where it's becoming interesting. So these are just some of the processes that are upregulated. You see, upregulation is a little bit more complex than the downregulation. Matrix remodeling, cell matrix adhesion, all of these are processes that normally happen at this time, but are upregulated even further. And we have increased response to progesterone without actually having at that time progesterone receptor being upregulated in transgenic line. So it seems it's doing something that Wnt4 is normally doing downstream of progesterone, but in this case without progesterone. We have some of the genes that are associated and necessary for decidualization being upregulated, and some glucose utilization genes. We have asked where the proliferation might be downregulated, and have just done the immunohistochemistry with MKI67 proliferation marker, and we've done it actually in both genotypes. We don't really see the difference, as one often doesn't if it's not big enough just by using immunohistochemistry. But what we see is that proliferation seems to be at this stage limited to the epithelium. Hence, also the downregulation will be limited to the epithelium. So luminal epithelium. We also asked what the story is with glucose utilization, and what we see is that here on the right, these are transgenic uteri, and this is the wild type, that we have a much stronger mucus production in these mice. So all that you see in colored here. All right. And then, of course, we asked ourselves what the signaling pathway is that the Wnt is using, just to have a little bit more idea what is going on. And what you see here is just a scheme showing the 3 kind of major ones that we know, and with canonical being perhaps the most well-known pathway. What happens there is that normally beta-catenin is marked for degradation in the cytoplasm, and in the presence of Wnt, it's actually that degradation is stopped. Beta-catenin gets into the nucleus and starts regulating genes. And so we asked whether that's the case, and normally what you would see is here green fluorescent is beta-catenin, and you see wild type and transgenic mice, and again wild and transgenic in proestrus and estrus. In none of these there is actually transport into the nucleus, so we assume that it's probably not the canonical pathway that Wnt is using. Also, the genes that are usually regulated are not upregulated in transgenics. Where we do see a little bit upregulation is in CAMK genes, and these are actually the genes. No, sorry, here. These are actually the genes that are associated with this calcium signaling. I cannot say very much more what it's doing. All right. Okay. So of course the question then is, how do we go from here to the effect on pregnancy? And there's obviously a lot of work to be done. What we seem to have to the functions that we seem to see affected are all very plausible, very directly associated with pregnancy. But really important to note here is none of these phenotypes that we see are actually during pregnancy. All of them are in preparation for pregnancy. Okay, so they would improve implantation rather than having an effect on pregnancy. And for me, this was a little bit of a reminder that we sometimes, when we look for effects of our loci, think that they have to be found directly in the place that we that has a direct function. So either during pregnancy or and so that's not always the case, right? Often we have to look broader, and we'll still have effect later on. All right. But I want to also address two other aspects still. One is what underlies the mechanistic basis of antagonistic pleiotropy itself. What does this all have to do with endometriosis or with cancer? And the other is the translatability to humans. What can we really predict based on what we see in mice about the effect of this allele in human? So what I think that underlies the antagonistic pleiotropy is probably invasibility. So the effect of this allele on invasibility, because of course in a particular context that we are looking at, namely embryo invading into the maternal uterus, the way how you have to transform the tissue is similar to the way that the tissue gets transformed in the case of cancer. So this interaction between the uterus and the embryo, and the ability to invade, dependent on the niche, is very similar between cancer and implantation. And there is some indirect evidence to that. One is the evolutionary pattern of uterine invisibility, and that is that the species that have very invasive placentations are also species that are very prone to metastasis. So that's one part. And another piece of evidence is the interesting observation that when we plan surgeries for breast cancer, the outcomes or the ability for breast cancer cells to actually implant elsewhere depends on the phase of the cycle. So they depend. Note that our invisibility here depends on the high levels of estrogen, right? So that there is a dynamics across the cycle when this effect will be there, and so when you make a surgery, how do you avoid seeding the cancer and having implantation of a cancer is also dependent in the same way of the cycle. All right, and the second question is about translatability between the mouse, what we find in the mouse, and what we can say about humans. And I think the translatability between models that we use for research and a different animal depend, of course, on different species, depend, of course, on how what homology do we have between the tissues into species, right? What can we say about that? And just a very quick run through the evolution of pregnancy here. We know that the really pregnancy with implantation has evolved so without implantation first in the stem lineage, leading to marsupials and eutherians, and then so we have in that case a very short pregnancy. At least in the basal lineages, we have a very short attachment, and in the next moment that causes inflammation, and in the next moment the fetus is out, is expelled. And what happened then here on the way to eutherians is that that actually implantation happens, and one long pregnancy, longer or shorter, whatever, pregnancy is inserted here, and we have again implantation at the very end that causes that is associated with labor and expelling the parturition. Okay. At the same time, when implantation evolved and the pregnancy evolved, there was also a strong evolution. So we often talk about placental mammals, but placenta is actually not novel here. What's really novel here is the uterine cell, namely the decidualized cell, the decidualized cell that we have talked about already, and that is associated with very invasive placentation at the beginning. So very strong and invasive implantation. What you see here plotted on this phylogeny is the invasiveness of placentation. So we have a hemochorial placentation is the most invasive one. We see it here. It practically puts the maternal blood in direct contact with the fetal vessels. And it's been made less invasive several times in evolution, but it's really important. We have this novelty here with the decidual cells, and these cells I mentioned already are present. If they're not present in mammals, they are secondarily not present, let's say so. But in the ancestral mammal, they are present. What changed in the evolution is that some of the in some of the lineages, like here in primates, but also in some of the bats, and in spiny mouse, and I think it's in elephant shrew, right? We have this kind of transformation not only associated with pregnancy, but it happens also during non-pregnant cycle. Okay, so that is different, and that is also different between mouse and human. Okay, so that's one thing that makes it a little bit harder for us to compare non-pregnant cycle between mouse and human. All right. Later gestation is way harder to try to compare, and that's unfortunately not talked about all too much. But we have, if you look at the development, human development during this long 9 months pregnancy, you notice that it actually develops very strongly. And if you look at the mouse development, this is at birth. One could compare it to a very early stage of the human pregnancy. So we cannot really stretch mouse pregnancy to the same length as human pregnancy and say, okay, mouse late pregnancy is the same as human late pregnancy, and compare that. That's probably not going to work. So I rather think that we have a comparability of whole mouse pregnancy at the very beginning of the human, and the rest is there's just no homologue to it. Okay. So to just summarize that part, I think that the processes that are homologous and probably can be compared, one is the cyclic preparation of the uterus for implantation, the implantation itself, and decidualization as a process, but knowing that it sometimes happens, that in human happens also in non-pregnant cycle. What we cannot compare probably is the direct regulation of pregnancy in later pregnancy. So we are, I think, pretty okay with our stage, and I think that the differences or the effects of our alleles that we are observing in transgenic mice are probably translatable. We haven't been able to use human tissue for that yet. You might know the fibroblasts are somehow very hard to CRISPR. We've tried. But so I hope we can have human tissue to actually look at the differences. The allele frequencies are high enough that just by sampling we should be able to get the right genotypes. All right, just to summarize the whole talk today, the immediate what, when, and where of the SNP effect, I think we have at least in mouse figured out quite well. What I think is quite surprising is that the pregnancy length in this case is actually affected by something that acts before pregnancy even starts. So it's really in non-pregnancy, in the processes that predate fertilization even. What we seem to find is that the tissue changes overlap with what had been reported to enhance cancer spread, and really note that this is not about the cancer itself, the cancer cells themselves that have this effect, but the tissue that interacts with cancer and enables the invisibility. That's at least our conclusion. And what we seem to find here is that in this case of antagonistic pleiotropy, it appears that it's actually really the very same mechanism that is relevant for the associations with cancer as with or endometriosis, on the other hand, as it is with preterm birth. And with that, I would just like to thank, of course, all of you for your attention. And I would like to in particularly thank Caitlin McDonough Goldstein is a postdoc in my lab and has done quite a bit in this in experimental part of this project, and of course many others in the funding. Thank you very much.